Transcriptional unit. Just before this transcription, we discussed about the replication of DNA. In replication of DNA, both strands are involved in replication, but in transcription, only one strand of DNA involved. Only one strand of DNA is involved. Out of two strands of DNA, only one strand is involved. Which strand is involved? That is the 3' and 2, 5' and direction. This strand only involved. And 5 to 3' and direction not involved. What happens if these two strands are involved? Why the two strands are not involved in this transcription? Why only one strand is involved? For example, the two strands are separated like this one. One, that is the 3. It is the only strand involved in transcription. The strand involved in transcription called template strand or antisense strand or code complement. In transcription, only one strand of DNA involved, and that strand is the 3 to 5' in direction. That only DNA strand is involved in transcription. The strand involved in transcription called template strand or antisense strand or code complement. Complementary strand or the 5' into 3' end strand not involved in transcription, not produce the DNA or RNA. That strand is called coding strand or sense strand or code copy. Code copy. Why only one strand is involved, and why? What happens if the two strands are involved in the formation of the mRNA? For example, template strand that produces the one type of mRNA, one type of mRNA that produces the protein. That is the one type. Suppose another strand also involved, it produces the another type of mRNA that also produces protein. Another type of protein. Only we required only one type of protein. Other protein not required. So when two strands are involved, useful protein and not useful protein, two different types of proteins that are synthesized. So characters that are changes. Protein generally they are involved in the structure of the body. Characters are the structure of the organism that changes. It is one reason. And another reason, the two strands are not involved in the transcription. Suppose it produces this mRNA and this is the mRNA, both are complementary one another. So double-stranded mRNA that is formed. When double-stranded mRNA is formed, not involved in translation. The two RNAs that are produced by two strands, they are automatically complementary to one another. So hydrogen bonds are formed and the two mRNAs become the double-stranded mRNA. Double-stranded mRNA not at all participate in translation. So protein synthesis stops. So these are the two disadvantages when the two strands are involved in transcription. A transcriptional unit, the segment of DNA involved in transcription called transcriptional unit. Transcriptional unit. Complete DNA not involved in the transcription. Only a part of DNA involved in transcription. Only part of DNA that produces the RNA. The segment of DNA involved in transcription called transcriptional unit. A transcriptional unit consists of three parts: promoter, structural gene, and terminator. These are the two strands of DNA. Only this strand, 3' and to 5' end direction strand, acts as the template strand that involved in transcription. Now, with reference to the coding strand, we are representing the positions of the different structures. Promoter, that present at 5' end. That the 5' end of coding strand. We are taking the reference as coding strand only. To propose the position of the different structures, we are taking the coding strand as reference. So, promoter position 5' end of coding strand. Terminator, that present at 3' end of coding strand. No need to say coding strand, just 3' end. The 3' end means 3' end of the coding strand. So, position of the promoter 5' end, position of the terminator 3' end. The 5' end and the 3' end taken as the reference of the coding strand. And in between the promoter and the terminator present structural gene. So, structural gene present in between promoter and coding strand. So, promoter and terminator. Structural gene present in between promoter and terminator. In this region only mRNA that synthesizes. That means from this region, transcription initiated. Then what is the need of this promoter? Here, RNA primer that attaches. RNA primer is attached here. That initiate transcription. Now, so these are the three structures present in a transcriptional unit. Transcriptional unit, the structure or the segment of DNA involved in transcription, consists of three parts, namely promoter, located 5' end of this coding strand, terminator, 3' end of coding strand. In between these two, promoter and terminator present structural gene. Structural gene actually involved in the formation of RNA by transcription.